Hello everyone. The purpose of today's presentation is to explain RNA-Seq. First, what are the applications of RNA-Seq? RNA-Seq let us investigate and discover the transcriptome, the total cellular content of RNAs including messenger RNA, ribosomal RNA, and transfer RNA. And understanding the transcriptome is key if we are to connect the information in our genome with its functional protein expression. Next, we will explain the flow of RNA-Seq. This analysis is roughly divided into two steps. The first step is to prepare the sequencing library. The RNA are extracted from the two samples to be compared and subjected to sequencing. The sequence data is output as nucleotide sequence data using the sequencer. Next, this sequence data is analyzed. The output data is saved in a format known as a FASTQ file. The next step is to quantify the genes. The quantification of the gene is first. Mapping is performed and quantification is then performed. Mapping is a process of taking the sequence data from the previous section and applying it to a reference genome, which is a non-nucleotide sequence. This will allow us to compare gene expression levels between normal and muted cells. In fact, it is summarized in a table like this. The first column shows the gene name. The remaining columns contain counts for each sample you sequenced. This data is used for downstream analysis. In fact, we use an application called Terminal to analyze. There are various visualization methods available, but we will discuss PCA and heatmap in this article. This is principal component analysis, a multivariate analysis technique that synthesizes a small number of uncorrelated variables, called principal components, that best represent the overall variability from a large number of correlated variables. The distance in this plot represents the amount of variation in gene expression. This example shows that the wild type and K4DT types have similar gene expression variability than SV40. Here is a heat map Red represents high gene expression, and blue represents low gene expression. This allows us to compare the gene expression levels for each sample. There are many other advantages to RNA-Seq like this.